Hi, my name is Shadi Atia, professor at Liège University, and today's presentation is an introduction to experimental research. Well, this presentation is the first presentation of a series or a playlist on experimental research, and there will be following presentations that are going into detail. Well, who is the audience of today's presentation? In general, experiments are at the core of laboratory research. So this presentation is for postgraduates and scientists in general who are conducting or seek to conduct their research in the field of social sciences, life sciences, medical sciences, and natural sciences. In this sense, also mentors and supervisors are concerned and they are welcome to watch this presentation. Let's have a look at the different methods of research. We have actually two families of research, quantitative research and qualitative research. Qualitative research, we previously presented it in a full playlist. Today we will focus on the experimental research as part of the quantitative research methods. Well, there is another approach that combines the quantitative and the qualitative. It's called the mixed approach. And in this sense, the researchers have to combine different techniques and ways or types within the four types of research. It's very important if somebody asks me how to distinguish experimental research from observational research, because many researchers are confused about the difference between observational and experimental. I would say that experimental research is manipulative, which means that we have the study design alters variable or variables for comparison against a control condition or a baseline. While if we talk about observational research, it is non-manipulative. We do not have a treatment. We do not interfere to change something in the natural environment that is observed. And the study design, that it is considered as the study design that typically represents realistic conditions. So this is very important to take into account. Another clarification when you talk about experimental research, in this series I'm going to talk about experimental research that is controlled. So controlled experimental research. And by controlled, I mean here I mean eliminating or controlling other variables, and I mean controlling the setting and the environment of our investigation, and we have an experimental control. Now, if we look at different or other alternatives like non-random selection approaches where we have experimental research with non-random selection, with no experimental control, we do more comparisons. Then we call that quasi-experiments, and I'm not going to cover that in my presentation today. This will be more covered in the observational research playlist or series. So let's start with the objective of today's presentation. Well, the main objective of today's presentation is to enhance your capacity to conceptualize, design, and conduct experimental research, number one. And number two is to formalize your research study design based on best practices. The content of today's presentation is mainly, I'm going to talk about an introduction about experimental research, what it is exactly, a definition for it, what are the experiments conditions, how to design an experiment, how to prepare for it, what should be going when we do analysis, and what is the validation and quality assurance measures before reaching the takeaway steps. But take care that the presentation of today is an introduction, so I will go running fast over these topics, and later on in the following videos you can have more detailed presentation or content. Let's start with the presentation, or let's start with the introduction. Well, the first thing to take into account when we are investigating or starting in the field of research, we need to be aware about epistemology. Epistemology, what is epistemology in fact? Well, it is the theory of knowledge, especially with regard to its methods. So actually, it is the theory of knowledge, especially with regard to its methods, validity, and scope, and the distinction between justified belief and opinion. So actually, it is the approach to learn about how to do the research. So it's very important to learn about the epistemological approach in your field, in your domain, whatever institution you are associated with, what is the historical epistemological approach of your research institution or research lab. Another important thing we have to look at here when we talk about experimental research, a very important term, which is the research design, which is how you designed your research and how you are taking steps and what are the elements of your research. And here's the definition is saying that research design is the overall strategy that you choose to integrate the different components of the study in a coherent and logical way, thereby ensuring you will effectively address the research problem. It constitutes the blueprint for the collection, measurement, and analysis of your data. So as you can see, any research design must include or must answer those four aspects. What is the data that will be collected? What will be the data that will be processed? What is the instrument or the variable that we are trying to analyze, understand, or even interpretation we are looking to interpret? And finally, how we can apply it and validate it later on. So those are the four major steps in any research design that you need to address and you need to answer when you are describing your methodology or designing your research. Now let's go further focus on experimental research. In fact, what is experimental research? Experimental research, simply the main purpose of experimental research is to contribute to knowledge by establishing evidence-based relationships between variables. Today we are seeking high evidence, we are seeking accuracy, we are seeking confidence in information or in knowledge and science, and the experimental research is allowing you to establish an evidence-based relationship between variables. Also, an experiment is a study of cause and effect. So the cause and the effect is strongly present in experimental research. It differs from non-experimental method in that it involves the deliberate manipulation of one variable or more while trying to keep all other variables constant or under control. So this is very important to keep into account when we define experimental research. We should not forget also that experimental research has a specific nature. So what is the nature of experimental research? As it is a systematic inquiry to describe, explain, predict, and control the observed phenomena. This is important, and also experimental research entails prediction, controlling, confirming, and testing. So in this sense, we are talking about integrity, research ethics, we are talking about reproducibility, we are talking about eliminating the impact or the influence of external factors and controlling external or extraneous factors. And for sure, it is structured and predetermined. So we must design it, and we must make sure that it is not random. We must make sure that it is not, let's say, done without a design, and that we can have a pre-test and that we have a post-test to make sure that what we measure or what we want to measure in relation to an intervention has been already documented before and after. So this is very important. Now one will ask me, okay, what is then the advantage of experimental research? Well, experiments are the only means by which cause and effect can be established. So this is the only way to have a solid confidence in the information or in the data that we generate, and we can really establish the cause effect. Also, it allows for precise control of variables, and experiments can be replicated. So in this sense, the more often an experiment is repeated with the same results obtained, the more confident we can be that the theory being tested is valid. So this is one of the highest approaches with accuracy and with robustness regarding the result and regarding the confidence. So data can be analyzed and inferential statistics take place, and these tests permit to assess the ability to generalize results. And one of the most important aspects we are looking at in the field of research is generalization. However, generalization is not easy because the levels of evidence vary. So have a look at this graph that is developed by the reference here. It is the levels of evidence, and it is developed by the Center for Evidence-Based Medicine. And as you can see, the lowest level of evidence is the expert opinion. In many cases we get confused. We go ask a medical doctor, or we ask an engineer, or we ask a psychologist about their opinion about a phenomena or a topic, or even a dietitian about a food or something, and we think that expert opinion is the highest level of evidence. It's not true. Actually, expert opinion is the lowest level of evidence. It is followed by group number 4, which is case series that are considered as poor quality cohort and case control studies. And then we move up to a higher level of evidence, which is mainly systematic reviews for control studies. Then we go to the almost the second best level of evidence, which are systematic reviews for cohort studies. Still, there is no homogeneous between these studies, but still systematic reviews for cohort studies like individual cohort studies, or low quality randomized control trials. Now, what is then the highest level of evidence and what is its relation to experimental research? It is the systematic reviews of random controlled trials. So if I have, for any phenomena I'm investigating, let's say I'm investigating the effect of drinking coffee on health or about longevity, then I have to do randomized control trials, several of them. Then I can conduct a systematic review, and if I find a common denominator or a common advice or a common recommendation, at that time I can only have a very high evidence, and I can make sure that this information is close to truth in the limitation of the age we are living, depending on the limitations of science. So it's very important to take this into account, and therefore the experimental research can allow you to conduct random control trials, and therefore we consider it a very important approach. If you want to know more about systematic reviews and randomized control trials, I advise you to have a look at the following two videos. Now, let's discuss or let's present the characteristic of an experiment. An experiment must be objective, it should explain, it should allow to test, it must have units, we must have a unit. So in this sense, when we talk about units, they are relatively large, and it must have variables. And when we talk about variables, we are talking about small number of variables that we are testing or that we are measuring, because it is so difficult to investigate the influence of a factor on another, or a variable, independent variable on a dependent variable. So in this sense, we have to eliminate or reduce as much as possible variables, and in this sense, if we have only one variable that we are investigating, that will be perfect. For sure, we can have multiple variables, but it complexifies the situation, because you have to have a certain randomness in your sampling, you have to have a certain replication aspect in order to have a randomized control trial, and you have to have a very strict control, and this is one of the most difficult, how to control the external factor and to eliminate their influence and make sure that only this independent variable has influence on the dependent variable. So the definition here is that an experiment is a quantitative research strategy that tests a causal relation between one independent variable and one or more dependent variables. So it's very important to take it into account. And if somebody asks me what's the difference between experimental research and the four other research methods, whether it's the modeling or the observational or the qualitative, as you can see here, observational research, the objective is to describe and to test. We use large number of units, which is the subject or the sample, and we have a big number of variables. While when we talk about experimental research, we aim to explain and to test, and here comes the cause-effect relation. We use relatively large number of units, and we have a small number of variables that we are investigating. When it comes to the qualitative, we are aiming to explore and describe and explain. We have small number of units and a large number of variables. So this is a very short comparison about the three methods of research. Now let's move to the following aspect. Experiments conditions. What are my experiment conditions? First thing, for my experiment, I must have a subject. What is my subject? It can be a human being, it can be an animal, it can be a plant, it can be a material, a structure, an object, a machine, a device, a prototype, a dummy. Whatever it is, I must have a subject, number one. Number two, I must have a setting where I'm conducting my experiment. And the setting is where the experiment is carried out. Many experiments are carried out in the laboratory. And this is the advantage here, where we can control where control can be exerted on the control variables, and thereby eliminating them. So here comes the importance of a lab, to neutralize the external variables and to only focus on the investigated variable. What else beside the subjects and the setting? Well, before going into detail, the lab type, we have actually different lab types, and depending on your field of investigation, you can have different lab types. You have, for example, engineering, energy-saving environmental labs. You have clinical labs, environmental labs. You have food and beverage, forensics labs. You have life science labs, material science labs, pharma labs, and for sure psychology and social sciences labs. Those are all types of labs, and depending on the field and the domain, you will have best practices and ways how to build these labs and how to conduct your experimental research within these lab settings. But again, let me remind you about something important, why we use or why we rely on labs. Because actually the labs will allow us to have controlled trials. And in this sense, we can eliminate or control other variables, control the setting and the environment, and here it is in red. Here comes the importance of having a lab, because I cannot do that in natural environment. If I'm doing a research and I'm investigating a certain influence of a certain parameter on a certain phenomena, most probably this will be observational research and not experimental. So it's very important to keep this into account when you are investigating or starting selection your research approach or method. Well, let's move to another aspect of today's presentation, the experiment design. What is an experiment design? First of all, we have a series of steps to go through to design our experiment. First of all, we need to know what is our research question. We need to formulate our main research question and sub-questions, which is considered as a crucial way. And in this sense, the questions need to be clear, focused, not too broad. They have a not closed-ended questions. They include research variables, and they must be analytical. In fact, there are 3 types of research questions. If you want to know about them, you can follow the videos on the video on the research questions. But in general, I need to have always a research question to start my research. In some cases, or in many cases, in fact, some researchers conduct their experimental research without research questions, but they rely on a hypothesis. And a hypothesis, this is the scientific method, begins with an observation which leads the scientist to ask a question. She or he then comes up with a hypothesis, which is a testable explanation that addresses the question. Also, a hypothesis isn't necessarily right. Instead, it's the best guess that we can have, and the scientist must test it to see if it's actually correct or not. And scientists test hypotheses by making predictions. If hypothesis is right, then it should be true. Also, then they do experiment to make observations to see if the predictions are correct, if they are the hypothesis supported. If they aren't, it may be time for a new hypothesis. So it can be positive hypothesis or negative hypothesis. So always when you talk about research design, you must have a research question and you must have a hypothesis, both or one of them. Well, now I'm ready. I know about experimental research. I want to learn about how to prepare my experiment. Well, before starting, one of the most important aspects you have to take into account, you have to have training in research, ethics, and misconduct. You need to know what are the common research misconducts, such as what is data fabrication, what is non-informed consent, what is plagiarism, what is image manipulation, the no permission for data information usage, data falsification, the use of animal or cruel ways of using animal, avoiding using animal, conflict of interest, non-disclosure and safety procedures, and so on. It's very important. There's a presentation you can follow if you want to go into detail, but this is the number one we need to learn about lab experiments. Then comes the pre-registration of our research project. It's highly recommended that we lay out our hypotheses, methods, and analysis in written before I start collect my data. This is a best practice. It shows that you will be consistent and that you designed your approach and that you will not react depending on the findings or depending on the results. So that's a best practice, and it brings credibility to your work. It brings a reputation, a positive reputation for you. It shows your commitment to a specific research approach, and this is a best practice. Now, what is next about these ethical concerns? The second thing when we talk about preparation, I'm done with my literature review and I did my operationalization. When we talk about literature review, I already conducted all the knowledge about the field. I know the concepts, I know the ideas, I developed a theoretical framework, and I am now ready to define my variables, and I determine the variables that can be measured for each concept, and I will even determine how can I measure these variables. So this is the part of the operationalization, and once I go through the defining of variables, I go through choosing variables. If you want to know more about operationalization and study variables, you can follow the previous video. Now, it's very important when I do my literature review on operationalization that I keep into account correlations. After finding several ways of operationalization, the concepts and turning the concept into variables, the data collection should start with 4 criteria in mind. The first criteria is to have set clear what are the variables I'm studying, what are the sub-variables I'm going to investigate, and what are the indicators I'm going to measure, and the units that allow me to measure them, and the tool that can help me to measurements. After that, I conduct an analysis of variance to check if there is a variance when I introduce my intervention or when I test my correlational approach. And if there is a variance, I can then start to seek testing or checking if there is a correlation or not. Sometimes there is a positive correlation, sometimes there is negative correlation, and in this sense we can investigate our cause-effect inquiry that we are from the beginning in the experimental research seeking to approach. And after that, only if I have a correlation, because it's not always like that. Sometimes you will investigate the variance, you will not find a variance, or you investigate and you find a variance but no correlation. But if there is a variance and then you check the correlation, you find it also positive or negative, then you can start to have a regression, and you can start with modeling, and you can examine the validity of your answer. So this is very important, but take into account that the hypothesis predicts a correlation that must be tested after data collection, not before. So the chosen variable must be tested for correlation that might lead to regression relation. And this is always not the case. Sometimes we do not succeed with that. So this is the second aspect after ethics and knowing, getting training about the misconduct. We need to conduct our literature review and operationalize, and to be very, very, very aware and go into detail in defining our variables and checking what kind of correlation we want to investigate. Number 3 in our research will be the measurements protocol. I need to create a study design or a research methodology, and researchers in this sense should seek to comply with normative standards and protocol. I need to know in my fields what are the common standards, what are the common protocols that are used, what are the terms, if there is a glossary, to use the same terms, the definitions, the procedures, what are the indicators, what are the metrics, what are the measuring techniques. And in this sense, I must comply with standardized methods and approaches based on community recognized protocols. So the number 3 that I have to think about when I do experimental research, what are the existing standards or protocols that are well established and considered as best practice in my domain and in my field of experimental investigation or experimental research, learn about them, be ready to apply them in my research. And in this sense, I just give here examples. It can be an ISO standard, it can be a CEN European standard or whatever other standard, but this is very important to have measurement protocols that I use as a basis for my approach. Number 4, as preparation for my experiment, I need to conduct the design of experiment. What is design of experiment? Well, it is the planning, conducting, analyzing, and interpreting the tests to evaluate factors that control one or more parameters. And design experiment is very effective because it's allow you to define the most important variables that you will test. Otherwise, you might be having a very tedious approach by testing all variables with their intervals, with their different ranges of intervals. So design of experiment is directly linked to operationalizing. It helps you to define the experimental units used in the experiment, and it helps you to extract the most important or the most effective samples for the treatment and control group to control the trials. So actually it's a very effective way, and there is in the presentation on design of experiment there is some example. But let me take you through this example. If I'm trying to conduct a test and build a correlation between the quality of a cake that I'm baking in relation to the taste, to the color, and to the consistency, I will have to define my different variables and I have to operationalize them. And as you can see, I will have for cooking this cake several factors: the oven, the sugar, the flour, the eggs, the ingredients simply. I have different levels of settings, the temperature, the cooking utensils, the ingredients quality, for example. And it will be very difficult to start to vary each of those variables. Otherwise, it will take me huge number of experiments to conduct my test and really establish a correlation. Design of experiment is a statistical approach that will help you to define the most important variables and combine randomly different matches between the different tests in a reduced number of tests. And this will be very effective and will make you advance fast without neglecting or without covering the concept of randomization. So the design of experiment is a systematic procedure that is carried out under controlled conditions in order to discover an unknown effect, to test or to establish a hypothesis or to illustrate a known effect. And here you need to define, when you do your design of experiment, you need to discuss your sample size, your sampling approach, your sampling representation, the independent and the dependent variables, and the cause effect. And the factors to be tested are the levels or the settings of those factors and the structure and the layout of experimental runs or conditions. And for sure, in the design of experiments, we try to seek techniques and methods that reduce the number of experiments, because it's impossible to do comprehensive number of experiments. It will be exhaustive, very expensive, and you might not end up your experiment within the frame of your project or your study that you are conducting. So design of experiment is so important because it will allow you to go to the essential, and it will allow you to select the most representative tests to study a certain correlation. Number 5, within our preparation, I need to define my boundary condition. I need to have my research questions and my motivation clear, but more importantly, I must define my inclusion and exclusion criteria. I'm very surprised that many researchers who conduct experimental research, they don't communicate their inclusion and exclusion criteria when they are describing their case study or their participation selection process or their cohort that they are working on. So it's very important to take into account boundary condition. You must know what are the system boundaries. It is a set of criteria specifying which system that you want to study from everything else, and it is useful to define things that are not part of your study. So don't forget about it, and also it will allow you to share and communicate your assumptions, and changing assumptions always changes the results drastically. So you are not allowed to change the assumptions during the course of the experiment. You must first have an assumption, terminate the experiment, report results, and then later on you can modify or update your assumption. So it's very important to look at the boundary condition. This is the 5th important element in the preparation of your experiment. What else do I need to do, and I still did not talk about when I'm talking about preparation for my experiment? Tools, equipment. I need to know what are the equipment I'm going to use. What is the history measurement techniques for this variable? How can I create the equipment? In many cases, people create themselves the equipment. If not, they will buy them. What training do I need for a specific piece of equipment? What kind of calibration I need? Is the equipment calibrated? How is it regularly calibrated? Is there a quality assurance company? Is there a contract, and so on? So these are very important aspects related to the preparation for my experiment. Then we move to another important topic, which is my lab notebook. The lab notebook is very important. It gives a permanent record of ideas, concepts, raw data, data analysis, observations. So actually this will be a backbone that you will use for documentation and for following up. And in this sense, the notebook is essential for every researcher because it will allow you to keep track of the evolution of the project. It will allow you to keep record of the chronological order of your work. It will help you to communicate with other researchers. It must have a physical trace of what you do, and it will help you to guarantee the originality of your research. And it is a piece of evidence to be used in the court of law if there is later on any problem with patent or with discoveries. So it's a so important document that many researchers in experimental research must have, and this is the only way to document everything, because it happens a lot that people forget how they did their experiment and what were the conditions, let's say temperature, content of the settings, and so on. So it's very important to keep into account. As part of the preparation and as parallel to my lab notebook, I must also start to do batch testing, which is simply laboratory testing procedure in which one test is done simultaneously on multiple specimens. I have to prepare myself, and I can do batch testing so that I get trained and I need to know about it. And also another very important thing is doing pilot tests. So the pilot tests are simply a trial collection data to collect weaknesses in the design and instrument and provide proxy data for selection of a probability sample. So pre-testing shall reveal errors and define the study design before the final execution. And this is definitely part of the preparation for conducting your experiment. Number 8, we move now to the running the experiments. Now I am, as part of my preparation, I need to apply the methods defined during the design of experiment. And what to keep in mind when running the experiment is that what is the type of experiment I'm conducting? What are the measurement tools that I'm used? Are the tools calibrated or not? I should take care of the ethical concern related to the experiment, the quality of the data gathered through the experiment, all the aspects. Those are the 8 factors or the 8 elements that I have to take into account, or my checklist that I should use when I am preparing my experiment. And this allows me to move to the next step, which is data analysis. Imagine I conducted my experiment. I am now ready. I have the results. What should I do with data analysis? Number one, comparative nature. I should take into account the comparative nature of experimental research. Usually, an experimental research compares variations of an experiment. So researchers fail in this process to measure against a benchmark. And this is very surprising. By default, when you do experimental research, you must follow a comparative approach, which means that you compare your intervention to the original state. And this is very important. So therefore, a statistically significant portion of subject in an experiment must be shielded from exposure to variables. If I have a sample, most probably I can divide it. One part of the sample will get exposed to the intervention, and one is not getting exposed, and then I must measure. I must measure before and after, and I must measure with the intervention and without the intervention. And in this sense, I can allow for comparisons. It's very crucial. Since there is a lot of room of error, for error within the scientific method, failing to compare is vital for accurate analysis. Very important key message. One common source of error within experimentation is the confirmation bias. Confirmation bias is a big problem with experimental research, and we try to avoid it by increasing the confidence level, like I mentioned, through randomization, through repetition, and through control. And therefore we say randomized control experiment or randomized control trials. It's very important in this sense. Once I have this in mind and I'm ready to compare and I designed my experiment for comparison, I can start running my statistical testing. Statistics for experimental research are simply a set of tools to analyze the collected data and to draw conclusion from it. So statistic for experimental research is helping in this sense, and it helps us also to validate our experiments. And in this sense, we try to seek to calculate the confidence intervals. We need to evaluate the accuracy of our results, and we will check if there is a variance, if there is a regression. No, first variance. After variance, we check if there is a correlation. If there is a correlation, we can check and go further with a regression and a cause-effect proof establishment. Now, one of the important aspects part of our statistical analysis will be our sampling, and the statistics in this sense means the type of the sampling, the list of the correspondents or the contact person, whatever are the subjects, are they a human being or not human being, the exact sample size, very important for each experiment, and the statistical test that you will conduct. These are statistical tests to establish to test the variance or for correlation. This is very important, or other type of modeling to predict. So it's important to define the statistical tests, and after that I have to describe all my covariates that are tested, and a full description of the statistical parameters, including the central tendencies, for example, the means or other basic estimates like the regression coefficient if I established a correlation, and variation for the standard deviation or associated estimation or estimates of uncertainty. And here comes the confidence interval, how many I can repeat the experiment, and what will be the variance or the difference every time I repeat the experiment, and what will be the difference between the results. So this is very important, and definitely it includes also the randomization. So these are important part of statistics. I have to look at them, but also I have to look at in my analysis to different aspects, like I mentioned before, the independent and dependent variable, the analysis of variance, the correlation, the regression and cause effect, or the causality if possible. Definitely it doesn't go in reality always like that. This is the ideal way. We will be very happy if we start finding a variance by introducing a treatment or an intervention for a variable. Then when we find this variance, we study if there was a correlation, negative or positive, and from there we can go with regression and causality. But in most experiments, it stops after variance. We find variance, but we cannot establish correlation. So it's very important to be realistic here, and design of experiment, it's a field of knowledge in the statistical world that works investigation, and it can allow you to do factorial design. So you have multiple variables with multiple variations and combinations, and it will have allow you to have the principle of component analysis. Now I'm done with the statistical analysis, and I'm done with the preparation, and I'm done now with my analysis. I'm coming to the last part of today's presentation, validation and quality, in relation to experimental research. Now, experimental research, we must always be concerned with the internal validity and the external validity of our results, and our ability to generalize the outcome. And in this sense, it's very important to come back to this graph, what I mentioned in the beginning, the level of confidence. And I need to remind you that the highest level of confidence is with randomized control experiments or randomized controlled trials, level 1, where we have a lot of variation, randomization, and control, these three aspects of quality. And in the same time, we have a lot of tests that investigated this phenomena, so that we conduct systematic review, which is very selective. And from the systematic review, we can trace a specific phenomena or a specific variable, and we can confirm or deny a certain recommendation, positive or negative, depending on the situation. So this is the highest level of confidence. So when we talk about experimental research, it is one of the ways that will give us very high confidence regarding a certain problem. Now, when we talk about confidence interval, this is the most important issue or the most important validation value. When we talk about any experimental research, we must communicate next to our finding the range of error or the uncertainty or the confidence interval. And in this sense, confidence interval is assigned to the value of an unknown parameter, and it's simply the degree of uncertainty around a certain effect, and it is including sampling, sample size, sampling errors, sample variations, and there is different confidence intervals, 90 degree, 95 degree, 99 degree, and this is the precision and accuracy regarding the experiments. If the experiment were to be repeated a large number of times, each time by randomly sampling the same number of individuals from the population, I can define my level of confidence. So this will be the variance of repeating every time, and then I can get my curve, and then it can define how much did the outlier affect the confidence interval. So this is very important. And here I want to wrap up almost with the 3R concept. In experimental research, we have 3 R's: randomization, replication, and reduction of noise or control. And those 3 R's must be always present when I'm doing my experiment. Let's say my subjects are humans. I have to divide my sample to be able to compare. I must make sure that I have a randomization. Replication also is very important. So we can see the first step is the definition of the population. Then I can have randomization and I have matching. Then I can go conduct my experiment and I can do technical replication. And if I have replication with randomization and there is a control or reduction of noise as much as possible, I can start to calculate my confidence. Also, it's important when we talk about experimental research that not every correlation means that there is a causality. which means that correlation does not mean causation. Correlation means only that there is a relationship or a pattern between the values of two variables. Between the two x, the x and the y, I can find a negative correlation or a positive correlation. However, causation means that one event causes another event to occur. So causation can only be determined from an appropriately designed experiment, and it requires a lot of further replication to make sure, and a higher level of randomization to make sure that really this correlation is not only represented in a descriptive way, but we are also getting to the bottom of it, that we are confirming that there is a causal correlation. And this is not always obvious with statistical analysis. So as you can see, the experimental research has a component related to the physical setting and to the design of your experiment, but also to the statistical post-treatment of the results and the depths of your treatment. Well, this is the end of today's presentation. Just very fast, some takeaway message in case you want to have a refreshing bullet points. Experimental research methods involve key steps: operationalization, design of experiment, and the preparation of the experiments, including all the ethical concerns, the pilot testing, and so on. There are necessarily tools that accompany the experimental research, mainly the lab notebooks, and the statistics. Very important to be prepared, need to have training about using my lab notebook, training about statistics, and also training about ethical concerns. They aim at establishing relationships between variables to compare. So experimental research in this sense is very comparative, and this brings me to the next slide, that there is a high importance of comparative approach. Placebo or baseline samples versus intervention samples is a very important and very common approach. Do your best to neutralize the effect of control variables so that you are sure that what you are testing or measuring is true, and there is no other external influence or extraneous factors that influence negatively your results and can be misleading when you are drawing your conclusion. Last takeaway, for every experiment there is a perfect human bias. Keep it into account. We are most of the time under the influence of our emotional brain, under the pressure to be productive, under the pressure of the funding limits, time limits, deadlines. We, in most of the time, can be easily biased. So just keep this into account. For every experiment, there is a perfect human bias. Try to avoid it. Be conscious. Avoid weak statistical tests. You must make good statistical tests, and you must measure your confidence interval, and you must communicate your confidence interval formula. That is, it's simply the mean of the sample plus the critical factor and multiplied by the standard deviation of your sample. This is very important to keep into account. It's simple. Research methods sections are very important when you are designing and when you are describing your experiment. So therefore, take time and make sure that the replicability can be possible, and you can include all this information in your methods chapter or your method section. Don't forget about the 3R principles: randomization, replication, reduction of noise or control. And as you can see, when we talk about reduction of noise, it's not only control, it's also including blindness. So we are talking here about blindness, reduction of noise, and randomization and replication, and we are always seeking in experimental research to establish a cause-effect correlation if possible. If you want to want more, you can find those videos, but by that I end up the presentation of today. Don't forget, I'm very excited today because this is the beginning of a playlist. This presentation is a beginning of a playlist on experimental research. There will be following videos that go further into detail, but with this presentation you have now an overall view about experimental research, what are the major elements that I need to be prepared to go through it, and I wish you luck with the next step. And I was today talking about experimental research and introduction. Thank you very much for your attention, and good luck with your future research.